और जब आप स्लाइड चेंज करते हो ना तो आपको नहीं बोलना है, तो आप एक सेकंड पॉज ले लो, चेंज कर लो और फिर आप बोल दो।
ठीक, क्योंकि वो आपकी कंटिन्यूटी
हाँ।
में ब्रेक होगा। ओके। Hello everyone. Today we'll be discussing nucleotides and nucleic acid chemistry. So before we get into the topic, let us see what all things we'll be discussing under this heading. One is nucleic acid. What is it made up of? Nucleotides, the structure of DNA, various types of DNA which are present in our body, and the RNA. So as we know that to understand a topic, we need to have a concept, and we also need to know what are the previous year questions which are frequently asked. So the frequent questions under short notes are nucleotides analog, DNA structure, the various types of RNA, and under long note there is a question on DNA molecular organization and the salient features and conformations of DNA. Now what is a nucleic acid? We all know that nucleic acid is polymer of nucleotide, and the most common nucleic acid that we study of is DNA and RNA. Nucleic acid, as I stated earlier, it is a polymer of nucleotide which is held by 3' and 5' phosphate bond. As you can see in the flowchart here that nucleic acid, it is a polymer of nucleotide, and nucleotide is made up of nucleosides and phosphate group. Nucleoside is further made up of nitrogenous bases and ribose sugar, that is, and pentose sugar. So nitrogenous bases are aromatic heterocyclic compounds which can be purine and pyrimidine. Purine is a double ring structure and it is adenine and guanine. Both these purines are present in DNA and RNA. Nitrogenous base pyrimidine, it is a single ring structure and only cytosine is present both in DNA and RNA. Thymine is present only in DNA, whereas uracil is present only in RNA. That was about the nitrogenous bases. Now looking at the biochemical structure of these purine nucleotides. So purine molecule, as we stated, one is guanine and the other one is adenine, which is guanine is 2 amino 6 oxopurine and adenine is 6 amino purine. While numbering these purine nucleotide, we do it in clockwise, anti-clockwise manner, whereas for pyrimidine, we do it in clockwise manner, as you can see here. Pyrimidines are cytosine, uracil, and thiamine. Cytosine molecular chemical name is 2-oxy-4-amino pyrimidine, and uracil is 2,4-dioxy pyrimidine. Thymine is 2,4-dioxy-5-methyl pyrimidine. 2 मिनट पॉज हो गया. So when we talk about the sugar, these are pentose sugar, which is D-ribose and deoxyribose. D-ribose is present in RNA, whereas deoxyribose is present in DNA. And DNA in RNA is differentiated on the basis of presence of these pentose sugar. So as we can see in ribose, in second position there is oxygen molecule present, that is in 2' position, whereas in deoxyribose there is no oxygen present. Okay, DNA contains beta D 2 deoxyribose, whereas RNA contains beta D ribose. Now the combination of the nitrogenous base and the sugar moiety forms nucleoside, and to this nucleoside there is a linkage. So in purine there is beta N9 glycosidic linkage, whereas in pyrimidine there is beta N1 glycosidic linkage. So now look at the molecular structure here, that at the N9th position of the purine molecule, the nitrogen atom is attached to the first 1' carbon atom of the pentose sugar. And in the pyrimidine structure you can see that first nitrogen atom is linked to the 1' carbon atom of the sugar molecule. Now coming into the various nucleotides and their nomenclature. So whenever there is a purine molecule, you will notice that it is ended up with the suffix "syn", whereas for the pyrimidine it ends with the suffix "dine". Okay. So when there is adenine, it is called as adenosine monophosphate. Similarly, for guanine, it is called as guanosine monophosphate. For cytosine, it is cytidine monophosphate. Similarly, for thymine and uracil. When it comes to deoxyribonucleotide, it is named as deoxyadenosine monophosphate, and for guanine, it is called as deoxyguanosine monophosphate. Similarly, for all other nitrogenous bases. Now, to form the nucleotide, it is always the composition in a way like nucleoside combines with the phosphate group. Now, these phosphate group which combines are basically ये edit करना होगा. Okay. So basically, nucleotides are phosphate esters of nucleosides, and these are composed of nitrogenous base, pentose sugar, and phosphate. And the esterification occurs at the 5th or 3rd hydroxyl group of the pentose sugar, and most of the nucleoside phosphate which are involved are attached to the 5' phosphate, and hence 5' AMP is abbreviated as AMP, but 3' variant is abbreviated as 3' AMP. As you can see the linkage here, that it is the phosphate group is attached to the 5' group of the sugar moiety. So when there is one phosphate group attached, it is called as adenosine monophosphate. When there is two phosphate group attached, it's called as adenosine diphosphate, and when there is 3 phosphate group attached, it is called as adenosine triphosphate. Now this was about the structure of the nucleotides, that is how from nitrogenous base sugar combine to form nucleoside, and the attachment of phosphate group leads to the formation of nucleotides. These nucleotides are essential and perform various functions in our body. So let us discuss about the functions of nucleotides. These are activated precursors of DNA and RNA, and ATP, as we know, is the universal currency of energy, and it is also required for intermediates in many biosynthetic pathway. It is carrier of methyl group in the form of S-adenosyl methionine. GTP is involved in protein biosynthesis as a source of energy, and it is also component of various coenzymes like NAD, FAD, and coenzyme A. These are also metabolic regulators, for example, cyclic AMP and cyclic GMP. Now, there's a central dogma of life. Central dogma of life tells us about the interrelationship between the 3 biomolecule, that is DNA, RNA, and the protein. So when there is a formation of DNA from the DNA, it's called as replication. When DNA to RNA is formed, it's called as transcription. Whereas when there is formation of RNA to protein, it is called as translation. Now look at this diagram. DNA to DNA is replication, DNA to RNA is transcription, and RNA to protein is translation. Whereas RNA to DNA is called as reverse transcription. So as we have discussed about nucleotides, and we know that nucleic acid, it is a polymer of nucleotides. So DNA and RNA are the 2 nucleic acid that we will study. DNA, it is a double-stranded molecule which forms base pair between strands. It is a genetic material, and the DNA which is bound, DNA in our cell is present in bound form to a protein which is called as histone protein. These histone protein along with the DNA forms chromatin. Chromatin condenses to form nucleosome, and these nucleosome is present in the nucleus of the cell. Now look at the structure here. This is a DNA which has major groove, minor groove, and This is polymer of deoxyribonucleoside monophosphate, which is covalently linked by 3' 5' phosphodiester bond. These base pair is connected by 3' 5' phosphodiester bonds, and there's a hydrogen bond which keeps the two strands together. DNA exists as double-stranded molecule in which two strands basically wind around each other to form a double helical structure. Now, under DNA composition, there's a rule which is called as Chargaff rule. So that states that DNA has equal number of adenine and thymine residue. That is, the number of adenine and the number of thymine is going to be the same. And similarly, the number of guanine and cytosine residue is going to be same. So Chargaff rule of molar equivalence between the purine and pyrimidine in DNA structure. That is, purines and pyrimidines will be present in the same quantity. Single-stranded DNA and RNA do not obey Chargaff rule. The DNA double helical structure was proposed by Watson and Crick in 1953, and the DNA, it is a right-handed double helical structure. It consists of two poly-deoxyribonucleotide chains which are twisted around each other on a common axis of symmetry. And these chains are in anti-parallel manner, that is, the 5' of one strand is paired with the 3' of the other end. So we know that the nucleotide structure has 5' end and the 3' end. So if one is running in the 5' to 3' end structure, the other strand runs in the 3' to 5' structure. Okay, so the two strands of DNA are complementary to each other. The spatial relationship between the two strands in the helix creates a major groove and a minor groove. What is the significance of these grooves? These grooves, they provide access for the binding of regulatory proteins to their specific recognition sequence along the DNA chain. So basically, these grooves are the binding site for the regulatory protein to which their specific recognition sequence is attached. And under therapeutic and the molecular research, it was found that, edit this. So there are certain anti-cancer drugs which are dactinomycin, that is actinomycin D, which exert their cytotoxic effect by intercalating into the groove of the DNA double helix and thus interferes with the DNA and RNA synthesis. The hydrophilic backbone, which is made up of deoxyribose phosphate of each chain, is on the outside of the molecule, whereas the hydrophobic bases are stacked inside the DNA molecule. So the overall structure of DNA resembles a twisted ladder. And the outer side is hydrophilic, whereas the inner part, which is bound to the other strand, is hydrophobic. The width of the double helix is 20 angstrom, that is 2 nanometer, and each turn of the helix is 3.4 nanometer with 10 pair of nucleotide, and each pair is placed at a distance of 0.34 nanometer. The two strands are complementary strands, as we can see that A pairs with T, G pairs with C, and they run in anti-parallel manner, as I stated earlier, that is 5' to 3', whereas the other one is running from 3' to 5'. And the two strands are attached to each other via hydrogen bond. So between A and T there is 2 hydrogen bond, whereas between G and C there are 3 hydrogen bonds. Now, out of the two strands of the DNA, one strand is non-template or coding strand, whereas one strand is template strand or non-coding strand. So it is the template or antisense or non-coding strand which undergoes transcription and leads to formation of RNA molecule. Now this was about the characteristic features of the DNA. There are various kind of conformations of DNA double helical structure which are present in our body. We'll discuss about those forms. So basically there are 6 conformations of DNA: A, B, C, D, E and Z form. And among these, B and Z forms are important. Predominantly B form is present under physiological condition, and A form is a right-handed helical structure and contains 11 base pair, and this tilting of the base pair by 200 away from the central axis. Z form, it is a left-handed helix and contains 12 base pairs per turn. Now we'll look at the conformations and the difference between the 3 conformations, that is A form, B form and the Z form. A and B are right-handed helical structure, whereas Z is left-handed helical structure. Among these, A is widest and Z is narrowest. If we talk about the plane of the base, three forms are perpendicular to helix axis. The central axis in A is 6 Armstrong holes along the helical axis. B has a tiny central axis, and in Z there is no internal space. The major axis is narrow and deep in A form, wide and deep in B form, and there is no major groove in Z form. Whereas in minor axis in A form is wide and deep, in B form it is narrow and deep, and in Z form again it is narrow and deep. Now look at the diagram showing B form of DNA, A form of DNA and Z form of DNA, and then there is another form which is triple helical DNA structure, in which there is a third strand which is found. So triple stranded DNA formation, it occurs due to additional hydrogen bond between the bases. Thymine can selectively form 2 hexene hydrogen bonds to the adenine of the AT pair to form what? TAT. That is, one adenine will form hydrogen bonds with 2 molecules of thymine. Cytosine can also form 2 hydrogen bonds with the guanine of GC pair. So one guanine will pair with 2 molecules of cytosine. Triple helical structure is less stable than double helical structure, and due to 3 negatively charged backbone strands in triple helix, it results in increased electrostatic repulsion. So this was about the triple stranded DNA. There is 4 stranded DNA. So basically a polynucleotide with high content of guanine can form a tetrameric structure which is called as G quartets, and these structures are planar and connected by hexene hydrogen bonds. Antiparallel 4-stranded DNA structure is referred as G tetraplexus, and the ends of eukaryotic chromosome, namely telomeres, are rich in guanine and forms G tetraplexus. This is the diagram showing the 4-stranded DNA. Now coming into the important part of DNA, that is DNA denaturation. So during DNA isolation in laboratory, we separate the 2 DNA helical structures. So for that, the first step remains is the denaturation of DNA once the DNA has been separated from other cell organelle. So as we know that the 2 strands of DNA are held together by hydrogen bonds, and the disruption of hydrogen bond, which could be because of change in pH or due to increase in temperature, it results in separation of strands. And the phenomena of loss of helical structure of DNA is called as denaturation, and the phosphodiester's bond are not broken by denaturation, and this denaturation is measured by absorbance at 260 nanometer. So now when we talk about the 2 strands of the DNA helical structure. They are attached to each other by hydrogen bond. So in denaturation, it is the hydrogen bond between the AT and GC that gets broken, and hence the two strands of DNA is separated as single strand, and this happens because of change in pH or due to change in temperature. As we can see that this is the double helical structure. Once it is heated, it leads to denaturation. The two DNA strands are separated. They are now single strand. Once the temperature is cooled down, it again forms the double helical structure. So hence there is again formation of the hydrogen bond between the two strands. Now coming into the melting temperature. So basically it is defined as the temperature at which half of the helical structure of DNA is lost. GC base pairs are more stable than AT base pair because there is 3 hydrogen bond, whereas in AT there is 2 hydrogen bond. Tm is greater for DNA with high concentration of GC for the same reason. Formamide destabilizes hydrogen bonds of base pair, and it is used in the recombinant DNA technology. Renaturation is also called as re-annealing, and it is a process in which the separated complementary DNA strand can form a double helix. That was all about the chemistry of nucleotides and the DNA. Now we'll be looking at the chemistry and the structure of RNA molecule. Thank you. Hanif? कितना टाइम लगा?
नहीं, मैं बोलता हूँ